Good day, everyone. So today I'm going to be presenting on my final year project entitled Bacterial Diversity in Peat Soil from Different Land Uses. My name is Anisia Benedict Bakar 64643, and my supervisor is Dr. Samuel Lihan, and this is my final year project presentation. So these are the things that I'm going to be talking about throughout this presentation. And let's start off with the introduction. So the Earth's biodiversity is majorly represented by microorganisms. Although we might not see it, they are literally everywhere. Soil is the home to the largest, diversified, and resilient microbial community on Earth. These microorganisms play different roles in maintaining the ecosystem in that area. So the microbial communities in the soil are greatly affected by both biotic and abiotic factors. So an example of a biotic factor is the organic matter, while an example for abiotic factor is temperature. So peatlands only cover a small portion of the globe, which is only 3%, and Sarawak constitutes 69% of the total peatlands available in Malaysia. However, these natural peatlands are continuously being cleared to make room for agricultural plantations. Researchers are applying molecular methods to further study this microbial community in the soil. So my problem statements are: a large portion of the peatland microbial community is still not known. Although there are studies, there are still insufficient amount of studies, especially on the microbial community of the land that has been converted to other land uses. So these are my objectives. So moving on to my literature review. So the first point in my literature review is the roles of bacteria in peat soil. As I said before, these microorganisms play different roles in maintaining the ecosystem in that area. So it is very crucial for us to understand what it is that they are doing underground. So the first one is the first process that they do underground is the carbon cycle. So not all microorganisms, soil microorganisms are involved in the carbon cycle, but only those that can produce extracellular enzymes such as the hemicellulose, cellulose, and lignins are involved in this process. And the main enzyme that are responsible to reduce the atmospheric carbon dioxide is the ribulose 1,5-biphosphate carboxylase oxygenase, or rubisco for short. So microorganisms such as the cyanobacteria, proteobacteria, firmicutes, and acidobacteria contribute to fixed carbon to the soil carbon storage. So moving on to the nitrogen cycle. Atmospheric air contains 78% nitrogen, and nitrogen is a crucial component of our nucleic acid and proteins for living things. Yes, like I said before, we are a living thing. We are living things. Nutrient cycle involves the decomposition, assimilative and dissimilative, and abiotic processes. And microorganism-mediated pathways in nitrogen cycle are the nitrogen fixation, nitrification, denitrification. Diazotrophs are a group of bacteria that are responsible in fixing the atmospheric nitrogen to ammonia before it will undergo nitrification and be oxidized into nitrate, then nitrate. Denitrification is done by denitrifying bacteria, and 10 to 14% of the bacterial population in soil is occupied by denitrifying bacteria. So the end products of denitrification are nitrous oxide and dinitrogen, which will go back to the atmosphere. So the next point in my literature review is the important factors affecting diversity of microbial community in peatlands. So the first factor that affects the microbial diversity is the pH level. pH level alters so much in the ground, such as the concentration of ammonia or nitrate. However, acidophilic ammonia oxidizing archaea such as Nitrosotalia devanatera ND1 and Nitrosotalia sinensis ND2 are actively performing nitrification process in acidic soil. Dominant soil bacterial groups, such as the actinobacteria, acidobacteria, bacteroids, and proteobacteria are not affected by geological locations. However, they are greatly affected by soil pH. The next factors that affect the microbial community in the peatland is the water content. So an increase in water level due to permanent inundation releases many nutrients such as ammonium and phosphorus to the pore water. This makes the area to be richer in nutrients. So there will be an increment in microbial diversity. So the increase in water saturation will reduce the niche differentiation, which leads to a stronger interaction between soil microorganisms because of the homogeneity. And this eventually leads to the development of high tolerance to environmental stress. Moving on to the next point, which is the peatland ecosystem. So the formation of peat occurs over time. It occurs naturally over a long period of time, and peatlands contain a high concentration of organic substances derived mainly from the degradation of the plants. So it is an important ecosystem that helps with the regulation of the global carbon. So there are two types of commonly known peatlands, which are the fen and the bog peatland. So the fen peatland is nutrient rich because these peatland obtain their nutrients from the ground. And they have relatively higher pH compared to bog peatlands, which I am going to talk about later on. And this type of peatland is dominated by vascular plants such as this Carex species. This type of plant dominates the fen peatland. So the bog peatland is nutrient poor because unlike fen peatland, bog peatlands obtain their nutrients from precipitates and not from the ground. And bog peatlands have lower pH compared to the fen peatland, which is less than 4 pH. And it is mostly dominated by sphagnum mosses. Sphagnum mosses have the ability to retain nitrogen and other nutrients, which makes it a good plant to live in the bog peatland. So conversion of natural peatlands. This conversion of peatlands alter the structure and composition of the soil. There is only less than 10% of Malaysia and Indonesia's peatlands that are left untouched. Cultivations on peatlands can only be done once the original or native plants are removed by the means of deforestation or draining. So these activities, these deforestation and draining activities, provide enough aeration in the rooting zone for new crops to grow successfully. So the native peatland cannot really have bigger plants because of cannot hold bigger plants. So these two methods are required to make the peatlands to be able to hold bigger plants, because draining consolidates the peat so that the land can hold bigger plants in place, but it consolidates it makes it stronger in a way. And deforestation reduces the rate of litter decomposition in the land. And my last point in my literature review is the detection and characterization of microbial community by using molecular methods. So in my study, I will be applying the 16S rRNA gene, which is commonly used as a marker for phylogenetic and taxonomic analysis, as they are highly conservative among different prokaryotes. And amplification will be done by using PCR methods. Fire sequencing can be used to sequence microbial genomes, which enables me to identify bacterial species and differentiate the strains without having to perform cloning. So my methodologies are site sampling, determination of soil properties, extraction of DNA and PCR amplification, fire sequencing data processing, and last but not least, statistical analysis. So these are the sites that I've chosen to collect my samples from. So the oil palm peat will be taken from Ulu Sebau, Bintulu. My rubber peat will be taken from Batu Lintang, Sri Aman. My pineapple peat will be taken from Ladang Nanas, Sungai Mata, Kota Semarahan. My paddy peat will be taken from Skuduk Paddy Field, Siburan. And these samples will be frozen until it is needed for microbial DNA extraction in probably March. Okay, so moving on to the extraction of DNA and PCR amplification. So the amplification will be done by using the ITS-1F, which contains the 454 pyrosequencing A adapter and a 6BP barcode, and ITS-4 with 454 pyrosequencing B adapter. So 2 microbial primers are going to be used to generate 16S rRNA gene fragments of 500 base pairs. Thermal cycling are going to be done throughout the process of DNA extraction and amplification. PCR amplicons obtained are going to be sequenced with this 27F bacteria primer. So once we obtain the pyrosequencing sequence, a trimming procedure will be done to get rid of non-16S bacterial reads. And by calculating 97 similarity cutoff, the sequences are going to be grouped accordingly by their operational taxonomic units, OTU. So one representative sequence will be taken from every phylotype to be blasted, and with the help of Silva rRNA database, the taxonomic identity of the phylotype can be identified. So for the statistical analysis, fungal and bacterial community are going to be visualized by using the principle of coordinate analysis based upon their corresponding abundance of OTUs. Formonova is going to be used to find the variance in the community structure. Determination of the minimum significant difference will be done by using the Mann-Whitney analysis. Linear discriminant analysis, LDA, together with linear discriminant analysis, effect size, LEFSE, are going to be applied to the diversity of fungal and bacterial taxa between sites. So these are my expected outcomes. The first one is the successful identification of microbial species from every land use. The next one is that I obtain the common dominant phyla such as the acidobacteria and proteobacteria can be found across all peat samples because they are, from previous study, they are known to be the common dominant phyla among all land uses. Next, the decomposition of properties of the soil, such as soil pH and water content, are expected to be varying from one another. So we expect the composition of the soil to be varying from one another, and they are not supposed to be different because they hold different types of agricultural plants. And hopefully the results obtained can be used for future reference by researchers and scientists. So my Gantt chart starts from October and ends in June. So the data collection will be done from October to December, and the proposal writing has been done from November to January. And sample collection are going to be done in February and March. Lab works are supposed to be done from March to May. And yeah, lab works are supposed to be done by then. Report writing and presentation will be done in May and June, and finally I will complete my final year project. Thank you. That's all from me. Hope to see you again next time. Have a nice day.